And the mastering things due
Sunday at midnight. Yep. Okay, so when we talk about the immune system, we can first talk about the way that we have barriers to prevent microorganisms from getting inside of us. So we can talk about really simply barriers to infection. Okay. And obviously the skin is a really good barrier, it being composed of dead epithelial cells, right? So when you get a burn or if you lose, part of your skin, you're very prone to infection, right? But the skin also has sweat glands. And the sweat glands produce water, but they also produce acid. And so you get what is called the acid mantle. So generally your sweat's a little bit acidic. It's usually like 4.5 pH to maybe 6.5 pH. So remember that 7 would be neutral. And so it actually depends upon what you eat. So I had this thing, maybe I mentioned it when we were talking about the integumentary system, but where I was eating tons of cherry tomatoes out of my garden, and I noticed that when I sweat, I would actually get a rash and it would burn, right? And it was like, oh my gosh, I'm taking in so much acid that I'm sweating it out, right? It was awful. So anyway, but acid mantle, the surface of the skin, is really important because that prevents bacteria from growing. It also prevents fungal infections. So you can get fungus on your skin and it can start to use the skin to grow, and then you would have to take a fungicide to get that off. So that is an important mechanism. So we also have the mucous membranes. So there is a substance called mucin, and mucin is a protein, and it is secreted by the cells, and it is a defense mechanism. It's a barrier to infection because it entraps microorganisms. So it's important because it can capture microorganisms, specifically when we talk about the respiratory system, right? So when we talk about the respiratory system, we'll talk about breathing through your nose and how important that is rather than being a mouth breather, right? So the nose is a filter. And so when you breathe through your nose, you have hairs, and then you have this mucous membrane that captures stuff before it gets into your lungs, okay? And then your lungs, the trachea also has mucus in it, which captures stuff, and then the mucus has to be moved back up the trachea, and then you have to cough it back up and swallow it. You generally swallow your mucus that you produce. Sometimes we cough it back up if we have an infection, and it has a strange color, then we might spit it out. Okay, so mucus and mucin is important. We also can talk about the stomach. So when we look at the stomach also has a mucous membrane, right, so the digestive tract. So this is in the respiratory tract and digestive tract specifically. Right, so the mucous membrane of the stomach is also protective. The mucus in the small intestine is also protective. We also secrete other things. So for example, we have an enzyme called a lysozyme enzyme. And lys means to break apart. And anytime you see the ime, that generally means, well, ace means enzyme, but ime also means enzyme. And so lysozyme is actually found in your tears and in your sweat and in your saliva. And so this is an enzyme that breaks down microorganisms. So it would digest bacteria, it would digest the fungi, right? And I just watched this really interesting thing that says the chemical, the chemistry of the tears is different depending upon why you are crying. So like emotional crying has a different, there's different chemical composition of the tears than if we're like tearing up because we have something in our eyes. So it's kind of interesting. The interesting thing is that the emotional tears have more proteins in them, And so they actually, when you're crying, they actually stay on the surface of the skin. So they move more slowly when you're crying than if you're like have an allergic reaction and you're like tearing up, or you have something in your eye, you're like tearing up. It's kind of interesting. So it's, they think that tears, emotional tears, have to do with, I need your empathy. Please pay attention to me, right? Or please be nice to me because I am crying. Yeah. We also have in our sweat, or we have chemicals in these, which are called defensins. And these are small antimicrobial proteins. So these are specifically in the sweat. We'll put in sweat. Okay, so all of these examples, these barriers to infection, these are all examples of innate immunity. This can also be called non-specific. The reason why it's called innate is when you think of like an innate behavior, that means it's something that is not learned, right? So what this means is that you are born with this, and it doesn't depend upon being exposed to a microorganism. Previous exposure, it doesn't depend upon it. So it's considered to be innate. So we'll talk next about specific or adaptive responses in a second. Okay, so these are innate responses. When we look at other mechanisms of innate responses, so I'm going to continue this down, innate responses, we also have what are called, what is called phagocytosis. And can anybody remember what phagocytosis is?
Eating.
Yes, so the cells are eating, right? So this is where white blood cells can actually go in and they can take in microorganisms and digest them. So what is an example of what is the most common leukocyte that you have in your blood? Neutrophils. Remember, never let monkeys eat bananas, that mnemonic device, right? So neutrophils are an example of a phagocyte. Okay, so this is a type of white blood cell, a type of leukocyte. It's different from lymphocytes, which we'll talk about. Okay, so I think neutro has to do with how it is stained. So that name doesn't really mean anything. So we also have large phagocytes, which are called macrophages. And so we're going to watch a video in lab, maybe this week, maybe next week, I haven't decided yet, that distinguish between neutrophils and macrophages. And macro does mean something, in that these are very large phagocytes that actually have the ability to send out little protoplasmic extensions, and they essentially lasso the microbe, and then they bring it back in, and then they take it inside the cell, and then they digest it. And what organelle do you think that they digest it with? What organelle in the body is involved in intracellular digestion? Lysosome, excellent. And so these use, right, use the lysosome, their lysosome, for intracellular digestion. Okay, so intra means inside the cell. Okay, so this is also innate. So one really interesting thing about it is that when we look at the difference between invertebrates, like when we look at those organisms that don't have a backbone, like we look at slugs and earthworms and spiders and insects, they all have this type of defense, right? So they all have They all have white blood cells, but then they don't have the specific type of defenses that allow their immune system to learn. And so it appears that that evolved when we became vertebrates. So vertebrates evolved the adaptive responses, whereas only invertebrates only have this type of response. There's been a lot of study looking at the immune system of sea urchins, for example. Oops, we already talked about it. Okay, so just ignore that. Okay. We also have other chemicals. So other chemicals.
These are also variants.
No. So these are phagocytosis was the other one, and then these are other chemicals involved. Okay. One other chemical involved is what is called interferon. And interferon, you may have heard of interferon therapy or as a medical thing. Interferon is a chemical that is produced. It's a small protein, actually. It's a chemical produced by cells that are infected with a virus. It doesn't protect the cells that produce it, but it actually is a signal. So it signals other cells that are nearby. So let's say you have this virus and it's starting to infect cells in your back of your throat. Well, those cells will signal the cells in the rest of your throat that there is a virus present and that they need to take up defenses. And so this signals nearby cells to produce antiviral proteins that inhibit the ability of the virus to enter the cell. This is still non-specific because it doesn't depend on, it's just any virus. Helps to inhibit their ability to enter the cell. So I think briefly they were using interferon, like if you had the really bad flu virus and they caught it early on. I think that they might have been using interferon in that way. So I have a handout, and then what I want to do is go over the diagram that's the second diagram on this handout. So viruses are not cells. So they're different from bacteria and fungi. Viruses are typically just protein and then genetic material. So they could just be protein and DNA, or sometimes they're retroviruses. So they're protein and RNA. You guys see okay? Should I turn on the light? Okay. So if we look at this diagram, so ignore the top one first, and we'll go to the second one. I skipped ahead, sorry. I don't know what my problem was. Antimicrobial proteins, there we go. Okay, so this is the diagram. So this is a diagram from your book, and so this is the virus. So the outer part of the virus is a protein. So this would be protein, and this is typically DNA. So notice how this particular virus has infected. So this would be the cell that is being infected. So the virus has actually gotten inside, and the way that the way that it reproduces is it actually puts its genetic material into the cell's genetic material, and it uses the cellular machinery to make a copy of that. But what this does is this signals the cell to produce an antimicrobial protein. So this is the DNA of the cell, right? This would be the nucleus of the cell. And this is what is called the mRNA. So that is just a piece of RNA that is then going to code for the protein. So we talked about that in cell biology. We talked about how messenger RNA can be translated into protein. So these are the antiviral proteins. Actually, this is the diagram of them right here, proteins. This is where they're pointing to them right here. So what is it called when a cell releases substances to its outside? Yes, exocytosis. So it secretes, or through exocytosis, it releases these proteins. And this is the flag. This is like warning, right? And so notice how then this protein can go over and bind to a receptor on the surface of a neighboring cell. So that's what that is. This is the receptor. So this is actually, let me write this. This is actually the interferon. Right, that's the, so those red triangles are the interferon. Okay. So this is the DNA of the neighboring cell, right? This is the messenger RNA. And these are the proteins that protect the cell from infection or block infection. So even if the virus does get in, it's not able to insert its genetic material, and so it cannot replicate. And so this can help stop the spread of infection. So this cell probably is still doomed, right? But the nearby cells might be able to survive the infection. So that would be an example of an innate response. Now, the other thing that we have in our body, besides the ability to produce interferon, are what are referred to as complement proteins. And there's about 20 of them. And typically, they reside in the blood. So 20 different kinds of complement proteins. And so one thing that these complement proteins can do is that they can enhance the immune response. So they complement the immune response. And so they can bind to foreign, they can bind to foreign substances, right, including microbes. It could be a toxin, right? So there's other things. Sometimes we get immune response to a toxin that's being produced by the bacteria, or a toxin that's been inserted in us, like venom or bee stings, right? So they can bind to the foreign substance and they can initiate its breakdown, right? So initiate the breakdown of it. So one example of a complement protein is a protein that is able to produce holes in bacteria or in foreign cells. And those holes inhibit the ability of the cell to maintain homeostasis. So fluid just gets lost, right? So this is called, let me see, I always spell this one wrong, opsinin or opsinin. So an example is opsinin. And this produces holes. And so this is just one example of one of those complement proteins in the bacteria or cell, other cell. So it could be a cancer cell, right? Or it could be a cell that's infected with the virus. So in this diagram, which you don't have a picture of, this is what this looks like. So you do not need to know the different pathways by which these complement proteins come together and the way that they are initiated. You just need to know that they come together and they insert themselves into the plasma membrane of a cell. This could be a bacteria. And then what happens is it just essentially, it's like a bullet, right? The cell will then just leak fluid to the outside, or the fluid will enter the cell and will cause the cell to rupture. Okay. So those are complement proteins. Okay, let me back up a little bit. Okay, so one example of how this kind of all comes together is what we call inflammation. And there are actually two different types of inflammation. So there's acute versus chronic. So when we talk about acute inflammation, it is like you have injured yourself, and immediately inflammation occurs where the injury has occurred. And it doesn't tend to last a long, long time. So it doesn't last like months or years, right? So that is versus what we call chronic inflammation. So chronic inflammation is not necessarily associated with an injury. It can be, but it is long-term. And it tends to be systemic. Which means it is not limited to the area of the injury. Right? So this can be a bad thing. When we talk about inflammation, we talk about it being a bad thing. This is what we're talking about. So this, they've discovered, is associated with different types of diseases. So it could be associated, for example, with cardiovascular diseases, specifically where you get a buildup of plaque in the arteries, like arteriosclerosis. So they think that it could be that you have an infection in your blood cells or in your circulatory system that starts the inflammation, and then you get inflammation in your blood vessels, and then that leads to plaque buildup, okay? So cardiovascular disease. They also think it's associated with autoimmune diseases, like Crohn's, like lupus. They also think it might be associated with obesity and diabetes and Alzheimer's. So they've kind of found that there's a link between obesity and diabetes, which we kind of talked about, I think, already. And then, maybe we haven't already talked about that. Maybe that was my other class. And then Alzheimer's, diabetes and Alzheimer's. They're actually calling Alzheimer's maybe the third type of diabetes because it's very similar to the diabetes where you have the cells stop responding to insulin. So insulin resistance. Anyway, so this is different from acute. So acute, right, is an adaptation. Right? It is a protective mechanism. It is limited to the area of injury. And it is short-term. Okay. So the inflammation has what are called the cardinal signs. And these include redness, heat, pain, and swelling. So when we look at the parts of inflammation, the first thing that has to happen is that your immune system has to know when tissue has been damaged, right? So we have signals. So we have chemical signals that signal that there has been damage to the tissues and that there needs to be an immune response. And so damaged tissue, so the first part is damaged tissue, produces chemicals that initiates a response called leukocytosis. And this is where specifically neutrophils that are stored in the bone marrow and in other parts of your body leave that area and they get into the blood. So you have an increased elevation of your white blood cell count. So this is where neutrophils enter the blood. Okay. The second thing is that the neutrophils are in the blood, but how do they know where to stop? Where is the infection? Where has been their damage? And so the process that this is involved in is that damaged cells in a blood vessel, the endothelial cells, near damaged tissue, and they actually call it sprouting. They sprout cellular adhesion molecules on their surface. So they put out these cellular adhesion molecules, and these have an acronym. They're called CAMs. Okay, so cellular adhesion molecules. So this causes the neutrophils to stop, right? So essentially it catches the neutrophils, and this process is called margination. So it's called margination. Okay. The third thing that has to happen is those neutrophils, which are actually quite large, have to squeeze through the capillaries and get into the interstitial fluid. And so this process is called diapetus. So diapetus is where the neutrophils squeeze through capillaries and enter the tissue. And then they follow the chemical trail. So the bacteria, for example, releases chemicals when it's in the tissue, and it's digesting tissue, and it's using your tissue as a source of nutrition for itself to grow and to divide. And those waste products actually attract the neutrophils. And so this is called chemotaxis. So the neutrophils are attracted to the foreign invaders. So they move towards them following a chemical signal. Okay. So those are the what happens during inflammation. And so if we look at a diagram on your handout, I'll go back to that in a second. Okay, so this is inflammation. So this one right here, where the neutrophils get put back into circulation, what is that? What was that called again? They get put back into, or they get passed into the circulatory system. Look in your notes. What was the first thing we talked about?
Oh.
When they make it through, you're asking?
No, so here is just showing them getting put into the circulatory system, so that's why that's called leukocytosis. Okay. When they adhere to the damaged endothelial cells, that is called margination. When they squeeze through, diapetus. Right? So this is just the capillary. This is the endothelial cells. This shows the neutrophils being attracted to the bacteria, so that is called chemotaxis. Okay. So when we look at inflammation, the reason why the neutrophils can leave the capillaries and other fluid actually leaves the capillaries as well is because the capillaries become leaky. And so does anybody remember what is the chemical that's produced by mast cells or basophils that causes capillaries to be leaky? Histamine. Histamine, excellent. Okay. So if we look back down here, okay. Did everybody get this one? I'm going to go forward then. We can talk about what is called the formation of exudate. Okay, so the exudate is what causes the swelling because it is fluid that is leaving the capillaries. And it contains some white blood cells, but also contains proteins. Okay, so we get histamine. Released from mast cells, okay, cause capillaries to be leaky.
I think this is happening to me right now.
You feel it? Okay, so you start to swell up. Okay. We also have, so this causes swelling. Okay. We also have an increased vasodilation of the capillaries in the injured area. We'll put blood vessels. I'll just put blood vessels because it could be also arterioles.
What causes the heat?
That causes the heat and the redness. And then we have chemicals that are produced by damaged tissue. And so this causes pain. So we have pain chemicals. Bless you. So the pain chemicals include kinins. Okay. So pain is interesting because it is adaptive, but we oftentimes don't want pain, right? But if we didn't have pain, we wouldn't know that we had injured ourselves. And so, you know, it's kind of interesting because as you get older, and if you think about really old people, you know, they'll like cut themselves and they'll be bleeding, you know, and they'll walk into the house and you're like, God, what happened, Grandpa? You're like, your leg's all bloody. And he's like, oh, I didn't even realize I did anything, right? And so it's this idea that the pain is adaptive because it allows us to know when we have injured our skin, right? And so it's important, but oftentimes we take drugs in order to counteract that. Another type of chemical that is associated with pain are the prostaglandins. So this is produced by all tissues, but it can be produced by tissues when we're, this can increase inflammation, but it also can cause pain. And so there are drugs that we take that oftentimes work at this level, specifically with the prostaglandins. And so what kind of drug do you take if you're trying to decrease inflammation and relieve yourself of pain? Antihistamine is inflammation, but what about pain? What kind of drugs? No, not gabapentin, because that works with them. No, don't take gabapentin. Do you take gabapentin?
No.
Okay, no, I don't. I'm a really anti-gabapentin person because I got put on that and it was bad. It's really bad stuff. So it is actually decreases the flow of action potentials in your nervous system, kind of shuts them down. But no, so like ibuprofen. So I don't take Tylenol, but Tylenol might work the same way. But ibuprofen, for example, inhibits the production of prostaglandins. So it decreases inflammation and it also decreases pain. Because the prostaglandins also work similar to histamine. They also increase. So ibuprofen would decrease pain and inflammation by acting on prostaglandins. Okay. Oops. So the last thing we're going to talk about before we get to the adaptive responses would be fever. So fever is actually an adaptation, right? It is an important defense mechanism.
So the bacteria kills what?
So your body can fight off the bacteria, yeah. So unless there's something wrong with you, or unless you're really little, so babies can oftentimes get overheated, and it generally has to do with the fact that if you put a baby in a sleeper and then you put blankets over them and you pile on the blankets, oftentimes they're not able to regulate their temperature as well as we are. So if we get hot, we just take our clothes off, right? When we have a fever, we get really hot, right? Fever is adaptive, but in children, mainly in children, it can get too high, and then it can be a problem. So what is the cutoff? Like 104? 104. So if it's over 104, it can start to cause seizures. Okay. So this is actually caused by leukocytes produce pyrogens. Okay, so the leukocytes have detected that there is a bacterial infection or a viral infection that's really bad. So they produce pyrogens. So what does that mean? Fire generating, right? So pyro means fired, and then gen means generating. So these are fire generating chemicals. So does anybody know what part of our brain regulates our body temperature? Hypothalamus, okay? So it produces pyrogens that travel to the hypothalamus. And cause an increase in the set point of body temperature. So your body is regulated homeostatically through a negative feedback mechanism. And so the set point goes up. So let's say it goes up and you were at 98 degrees and then it goes up to 101. How do you get your body, if it's set at 101, how do you get the body temperature to go up? How do you increase your body temperature when you get a fever?
Your heart rate?
Have you ever had a really bad fever?
Yeah.
And you go, ooh, you're like shivering, right? You can't stop shivering, right? So shivering, specifically, right? This increases the body temperature. You can also get under the covers, right, and shiver, or you could take a hot bath and you could stop to help increase the body temperature, okay? So shivering is a sign of the onset of fever. And then once your body temperature gets up high, then you will, up to where your set point is, then you will stop shivering, okay? When the fever breaks, what do? Sweat, okay? So this decreases body temperature. So, you know, you've been laying in bed and then all of a sudden you're like, oh God, all of a sudden I'm so hot and I'm sweating, right? And then that's because your hypothalamus has set the set point back down to where it was normal, right? And you have to get rid of the excess heat, okay? So this is onset, and this is where the fever breaks. Okay. So they do recommend that if at all possible, if you have a low-grade fever, that you do not take fever-reducing chemicals, because you might actually be able to get over the illness faster if you don't take fever reducers, right? But fevers make you feel really bad, and it's hard to function with a fever, but maybe it keeps you at home instead of going out in public and spreading the illness to everybody, right?
So I have a constant body temperature of 96.8, so I have like a low temperature always.
Mm-hmm.
Would that be because of my hypothalamus thing?
Are you always cold?
I don't feel cold. It
No.
feels normal.
So your body temperature, it's because your hypothalamus set point is lower. Yep. And it does lower at night. I don't know if you've discovered this phenomenon. When you go to bed, you're like freezing cold and you put everything on you, and then in the middle of the night, you're like, oh. And that's because your set point's gone down, right?
I'm always freezing. She's always hot.
Yeah.
I'm like turning purple. I'm like, I'm freezing. She's like, it's hot here. Turn the air conditioner.
Yeah. Okay. So that is innate. Fever is innate. Okay, so now we're talking about adaptive defenses. This is found in the vertebrates. Okay, so even snakes and lizards and fish, they have adaptive defenses. When we look at these adaptive defenses, these are also specific defenses. And so what this means is that they are, the immune system is able to learn, okay? So capable of learning to identify specific pathogens. That's why it's adaptive, it's not innate. And that's why it's specific pathogens. So our ability to detect specific pathogens has to do with the fact that they possess what are referred to, well, actually all cells possess what are referred to as antigens. Now, formally, this actually means that this is anything that can generate antibodies. So antigen, think about this as being antibody generating molecules. So antibody generating molecules. Okay. Now, antibodies are not the same thing as antibiotics. Antibiotics are chemicals that we have harvested from other organisms, like other bacteria and fungi, that inhibit cell division of bacteria. But antibodies are specific types of molecules. So antibodies, these are Y-shaped molecules that bind to antigens. So really, they actually have the ability to lock onto the antigen. So sometimes they say the antigen is the key and the antibody is the lock, and they fit very specifically molecularly. They kind of fit like a lock and key mechanism. So I'll put that on here. So this is the lock and key mechanism. Now, the adaptive responses are only initiated by the lymphocytes. So I mentioned the two different types of lymphocytes, and do you remember what they were called? B and T. Is that what you said? Okay, B and T. So B cells, right? Why are they called B cells? Right, they're produced in the bone marrow, but they mature in the bone marrow, and then they're released, okay? So the B cells are responsible for what is called humoral immunity. So humoral means fluid. So it used to be, the body fluids used to be called our humors, right? They would take a sample of body fluids and it would be called the humors. So humoral immunity generally means in the blood, right? So in the fluids of the body. So the B cells produce antibodies. So they are ultimately associated with the production of antibodies. We'll talk about how they do that. Okay. And then we have what are called T cells. And T cells are also produced in the bone marrow, but they mature where? Thymus, yes. So the T cells are responsible for what is called cellular immunity. And there are different types of T cells. The one that you might have heard of most is the helper T cell, because the helper T cell is the type of cell that is infected by HIV and what ultimately causes the formation of AIDS in people that have HIV infections. Okay, but there's also cytotoxic T cells, and there's other memory T cells as well. So there's different types of T cells. Okay, so cellular immunity, these specifically can attack cells body cells that have been infected with a virus or are cancerous. So we do have a mechanism to deal with cancer cells. Cancer cells typically produce weird proteins, and those proteins, those antigens, get put on the surface of the cell, and the T cells can detect them as being different from normal cells, and so that they can attack them and cause the cell to die. So lymphocytes are associated with adaptive immunity. Okay. So in your book, they just distinguish between these two types of these two branches of immunity. And so you'll notice that the B cells are produced in the bone marrow, right? But they can actually go into the lymph node. So remember, we talked about how lymph nodes are aggregations of immune system cells that can then be rallied if there is an infection. And then the thymus, the T cells can also end up in the lymph nodes. And then they can go into circulation, or they can specialize and start producing, B cells can specialize and start producing antibodies. So this is a diagram that just distinguishes between the B cells and the T cells. Okay, so before I want to talk about this one, I'll come down here. So we all have on our cells, on the surface of our cells, antigens that our body is able to use to tell our immune system that this is who we are. And so the immune system must be able to recognize self. Self from non-self. If it doesn't, what type of disease evolves? If your immune system doesn't recognize that these are your cells, then what happens? Autoimmune diseases, and those are really bad, and we can't even figure out how to cure them. They can only treat them. Okay. So in order to recognize self, we have a set of proteins that are called Mhc proteins. And this is short for major histocompatibility proteins. Compatibility proteins. Okay. So if you think about this word, histo means what? What's the study of tissues? Histology, right? So this is tissue compatibility. And so the reason why they're called this is this is what they discovered when they were trying to give you, like they wanted to give you a tissue transplant, or if they wanted to give you an organ transplant, your body would reject it because those proteins are going to be different from yours. So these are unique to the individual. Oops, individual. So I have different Mhc proteins than you do. The only time you would have same is if you are identical twins. So like it would be really good if you needed a kidney to have an identical twin, because they could probably donate it without a problem, right? So it's kind of interesting because these Mhc proteins seem to be also associated with the types of pheromones that we produce. And there is some evidence that we choose who we want to have children with based upon how different their Mhc proteins are from mine. So I'll be attracted to people that have a different type of Mhc protein so that my offspring will be more variable than if we had the same Mhc protein. So there's some evidence there that's kind of interesting. So these Mhc proteins are important because they, on the surface of the cells, so they're located on the surface of the cells, so they're located on the surface of every cell, and they bind, or they can bind or present antigens. So they can bind, I'll just bind and present antigens to T cells. So one of the things that's really interesting is that when T cells and B cells undergo cell division and they start to specialize, there is a chance that a T cell would be produced that would accidentally recognize the Mhc protein that you have as being foreign. And that might be a mechanism for autoimmune disease formation, right? And so we have the ability to select against cells that might accidentally attack normal cells. And so here you can see, this is my self-Mhc protein, and it has an antigen associated with it. If this T cell fails to recognize this body cell as being one of its own, then it will undergo apoptosis. And so there seems to be, when they go to the thymus, the reason why they're going to the thymus is that they're learning and they're being selected against if they are unable to recognize self from non-self. So in the thymus, T cells that might attack healthy cells, healthy normal cells, are destroyed via, it's essentially self-suicide, right?
No, it's just how depressing. It feels rejected. So
yeah, that sounds like called apoptosis. So oftentimes they say the T cells go to the thymus school for a bit of higher education, right? And the reason why is that you have to make sure that you're not going to pump out a bunch of T cells that are going to attack your cells and cause tissue death. Okay. So you don't need to know the difference between positive selection and negative selection. Here, they're just making sure that the T cells can recognize a foreign antigen that is on the MHC protein, and then it would be accepted. So here it is. Recognizing a foreign one, and then actually let's say failure to recognize. So this is the failure to recognize, sorry, a normal antigen, and so it dies. This one that recognizes a self-antigen does not die. If it does die, that could lead, or if it does not die, that could lead to autoimmune diseases. Okay. So this is what an antigen, this is a diagram of an antigen showing that it has different binding sites. Generally, antigens just have one binding site, but what they're showing here is the lock and key mechanism. So this is my antigen, and notice that it has a specific shape, and that's called its antigenic determinant. And then that Y molecule, the antibody, can actually bind directly to that antigen. Okay, so we'll talk a little bit more about humoral immunity. I have a video that we're going to watch in a little bit about the history of vaccinations and how this relates to humoral immunity. Okay, so when we look at a B cell, each B cell has only one type of antibody receptor associated with it, or antibody bound, we'll just put bound, antibody bound to its membrane. These antibodies are produced randomly, okay? So antibodies are produced randomly, and they actually think that the genes that code for antibodies get shuffled. They have lots of shuffling of the genetic code. This is important because we do not really know how the B cells could, by purposefully design themselves to specifically detect a particular microbe. So they think that it's just that there's so many random B cells generated that by just chance, they'll be able to bind onto an antigen if it is present, okay? So then we have selection of a B cell occurs if its antibody receptors can bind the specific antigen present. So that's just by happenstance. There happens to be a B cell that's been randomly produced that locks onto a bacteria that has invaded your body. And the selection then results in what is called clonal expansion. Clonal, what type of cell division is cloning? Is it mitosis or meiosis? Mitosis, right? So mitosis, so mitotic cell division. So that B cell that's been selected and is the right one then produces clones of it. And some of those clones specialize into plasma cells. And they're called plasma cells because what essentially they are little manufacturing plants that are then going to pump out the right antibody, the same antibody that was bound to that particular B cell, and then they're going to pump out those antibodies to the blood. Okay, so these produce antibodies that are released into the blood. So the diagram I gave you has a back sheet to it. This is the humoral immunity response. Okay. So what you can see is that each of those 3 B cells that were randomly generated have different types of antibody receptors associated with them. And so if you look really closely, you can see that there's different shapes. Like this one maybe has a U shape, right? This one has this type of shape, and this one has this type of shape, right? So the receptors have different locks on them, actually locks on them. Okay. So notice that this would be the microbe, okay, with a specific antigen. Okay. This is binding to B cell antibodies receptor. Okay. Cell division, right? This is clonal expansion, so this is mitosis. These are the plasma cells. Now notice that they have lots of rough ER in them because rough ER is where proteins that are going to be secreted to the outside of the cell are produced. And so you can actually see that when you look at them, that they're just full of rough ER. So they're essentially protein factories. So these are the plasma cells. Now this one is unique. This is what is called a memory B cell. It could stay in your body for years. It might even stay in your body for decades or maybe your whole life. Memory B cells could just could hang around. Okay, so this is the basis upon learning. So this memory B cell is going to be there if you are ever presented with the antigen again. So notice this. Notice that the B cell was there, and then the B cell was more quick in detecting the antigen, and that you produce a lot more antibodies the second time you are infected. So I could write first exposure up here, and I could put second exposure down here. And notice the second time, right? Even have more memory B cells. Plasma cells, these are called the antibodies that are released free circulating into the blood and can enter into the tissues.
Are these memory B cells part of the reason why if you don't finish your antibiotics that your body is like adapted so you would have
no
B cells?
So that is kind of confusing. Antibiotic resistance, which we'll actually talk about later, but antibiotic resistance doesn't work like that. Antibiotic resistance means that the microbes have adapted. You haven't adapted. The microbes have adapted, so they are now resistant to the chemical. And so yes. So this is the basis for vaccinations.
But what it says is you have to finish your antibiotics. Have you ever been told
Right.
that? Or
yes, and the reason why, if you don't finish it, then you have only killed off the microbes that are easily susceptible. And then what you've done is you've left the population of bacteria that are a little resistant. They're going to reproduce and produce more resistant, and you're going to get resistance building. Yeah.
I was just wondering if you know, like, how like chickenpox can morph into shingles, if that has any relevance to this.
Yes, when we talk about vaccines, we'll talk about that. The virus can actually hide out. So viruses can hide in cells, and if they hide in a normal cell and they don't do anything and they're just kind of dormant in there, then your immune system can't find them. And it only is when you're stressed out that generally that you get shingles, but we'll talk about that too. Okay. So this is the basis for humoral immunity, and it's also the basis for vaccinations. So if we look at the first exposure to an antigen, so this could be a vaccination, or this could just be that you get sick. So let's say that you are exposed to the flu virus, right? You start to produce antibodies, but there's a period of time where you just feel really bad, right? And notice how it takes a little while for your antibodies to get in there. So this is like the cold, right? You're like, oh, after a week, you know, I'll feel better, right? And that's because after a week, you start producing the antibodies. Now, if you are exposed to the exact same virus, cold virus again, you might feel sick for a day, but what will happen is that your immune response will be so much greater that you will not get fully sick, right? So the problem with cold viruses is just that they're constantly changing. So that's why you can get a cold and you're like, oh, I just got over a cold, and then you get another one. It's because you got a different strain and it changed over time, okay? But notice that this one, if it's a different one, then you still have that lag time. Now, when you go in for a vaccination, when you look at a vaccination, this second shot is often called what?
Booster.
A booster. You're like, why is it called a booster shot? Now you know, because it boosts the production of antibodies, right? And it actually makes your memory more stronger because you have more memory cells. So if you have a particular vaccine where you have to get a booster, I think the HPV is one of those, the human papilloma virus they're now having children get. I think they want you to get 2 or 3 of them because what they want you to do is to develop lots of memory cells so that if you're ever exposed to that virus, you'll be able to respond really quickly to it.
So what is it about the shots and stuff that people are concerned about? Like, what are the side effects then that people are always arguing?
Well, we'll talk about that, but there's lots of controversy because when you put something foreign in your body, there's always the risk. There's always the risk that you are going to have an allergic response to it, and you could go into anaphylactic shock and you could die. Yep. That's why they always have the anaphylactic, when you get vaccines, you know, they always have the shock cart right
I
there.
haven't had one since I was a kid.
Yes. So they want you to take the vaccines so that you can, they're prepared for you to have an allergic response.
Wow.
So, but we'll talk about the benefits of them. And I think unfortunately, I think my video might be a little bit too long for. Oh, it's 5 minutes. Do you guys have time to watch this video on vaccines? Yeah, sure. Okay. And we'll talk more about vaccines in 5 minutes here. Oops, wrong one.
It's hard to imagine a smallpox outbreak today, but for centuries, the fatal disease plagued mankind, wiping out entire populations. Thanks to the advances of vaccines, the virus was eradicated by 1980, and today only exists in freezers. The virus is believed to have appeared around 10,000 BC. Its earliest known victim was the Egyptian pharaoh Ramses V, whose mummified face shows skin lesions that indicate he suffered from the disease. A case of smallpox started off with flu-like symptoms, then a rash, blisters, then scabs. If it didn't kill its victims, it often left them horribly disfigured and sometimes blind. But 2 observations contributed to the discovery of vaccines and ultimately the end of this terrifying disease. First, people saw that smallpox survivors became immune to it. And second, people noticed controlled exposure to the disease led to a milder outbreak. Around 1000 AD, Chinese doctors began grinding smallpox scabs into powder, which they blew into the noses of their patients. In India, around the same time, physicians would dip sharp needles into the blisters of smallpox victims and then pierce the arms and foreheads of their healthy patients. These forms of inoculation, known as variolation, eventually spread to Europe. By the end of the 17th century, Europeans were known to send children to the homes of recovering patients to purchase crusts from their scabs for a penny or two. Children were also sometimes bedded with other children so they could contract smallpox under favorable conditions. Lady Mary Worley Montagu, an aristocrat, is widely credited for introducing variolation to England in 1721. After she had both her children inoculated, variolation became a regular practice and eventually reached the New World. But variolation had its risks. The inoculated patient would develop severe rashes, and they could spread it to others. But the worst side effect was death. As an 8-year-old in England, Edward Jenner was inoculated through variolation. Later, as a physician, he heard a milkmaid say, "I shall never have smallpox, for I have had cowpox." Her words sparked an idea. And in 1796, he found a young dairymaid, Sarah Nelms, who had fresh cowpox lesions on her hands and arms. Using material from the dairymaid's hands, he inoculated an 8-year-old boy, and then inoculated the boy again, this time with smallpox material. The result? No disease. Jenner called this new method vaccination, after the Latin word for cow, vacca. His vaccination protected against smallpox without causing severe rashes or exposing others to the disease. Eventually, vaccination replaced variolation. Whether they knew it or not, Jenner and those before him were mimicking the body's natural defense against disease. Our bodies recognize foreign invaders, such as viruses and bacteria. These organisms carry markers on them called antigens that trigger the production of disease-fighting antibodies. Once these antigens are destroyed, the cells that attack them retain a memory of the battle. The next time the antigen attempts to infect a person, these memory cells fight back. This protection is called immunity. Vaccines contain the same antigens or parts of antigens that cause diseases. Only the antigens in vaccines are either killed or weakened. They aren't typically strong enough to produce the symptoms of the disease, but they are strong enough to get the immune system to produce the antibodies. The memory cells that remain prevent reinfection. In 1967, the World Health Organization launched the successful global vaccination program, and smallpox was eventually eradicated. Jenner's first vaccination helped spur a whole new field of medicine called immunology. There are now 27 vaccine-preventable diseases, and scientists continue to work on developing new ones for diseases like HIV. Some challenges surrounding vaccinations include making them available globally, as well as an ongoing debate about their possible side effects. Despite these challenges, one thing is certain: vaccines have helped save millions of lives. For the Wall Street Journal, I'm Christina Suey.
There you go.
So the interesting thing about smallpox that they didn't talk about is that the Europeans brought it to North America, right? And it decimated the Native American population. It was very, very bad. They also brought it to South America.
Lab on Friday?
Lab on Friday, yes, 10 o'clock. ST-119.
It's 10 o'clock? Yeah. It's always at 10. She said 9 o'clock. I was like, 10 o'clock?